Hello and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Francesco Loria. So, listeners, Francesco is a PhD student in biomedical science at the Swiss Laboratory for Doping Analysis in Lausanne and the University of Geneva. And today we're excited to talk about Francesco's career, his research, and his work that led to the receipt of one of this year's PCC-sponsored anti-doping predoctoral awards in partnership with the American Physiological Society. So, Francesco, we're excited to welcome you to the show today. How are you?
Hello, everybody. Thank you for having me. I'm fine today.
We appreciate you spending time with us today, and we're excited to learn more about you and your work. So, can you start by telling us a little bit more about yourself?
Okay, so as I said, I'm doing my PhD here at the Swiss Laboratory for Doping Analysis. In my previous study, I've studied biology and medical biology at the University of Lausanne in Switzerland. And now I'm doing my PhD thesis about anti-doping and more about blood doping and biomarkers to improve the blood doping's fight and be better in anti-doping.
Phenomenal. Well, I'm excited to talk about some of the details of your research, but I'm curious. I know everyone sort of finds their way to anti-doping through a different path. So what was your path, Francesco? How did you first become interested in physiology and anti-doping?
So I started being interested in anti-doping because by my big passion about sports. And for me was the ideal field to combine my passion for biology and also my passion for sports. And also physiology. I discovered the physiology world during my university studies. And when I heard about PCC and the collaboration with APS, I go more inside physiology.
Absolutely. So then how did you find your way to the Swiss Laboratory for Doping Analysis in Lausanne and the University of Geneva for graduate school?
I come here during my master study. So I did my master internship there. And then I have a 9 months project here. And then I stay here for my PhD.
Now, you mentioned briefly about the PCC, but let's dig into a little bit of detail here. What is the story behind how you found out about the PCC and maybe how you decided to apply for this American Physiological Society PCC-sponsored research award?
So I heard about this grant last year when it was at the old Congress, the Experimental Biology Congress. And I applied also 1 year ago for the Experimental Biology. And then this year I apply again for the new APS Congress, the new format. And I'm grateful because I won the grant this year.
Well, congratulations, of course.
Thank you.
You're doing some really phenomenal research there. Your research is focused on developing biomarkers for blood doping. I think this is a particularly interesting topic. So to give our listeners some background, Francesco, what are some of the physiological changes that occur after blood doping?
So after blood doping, what happened? That the athletes used to have this kind of doping to improve first of all, their resistance. So it's more about the cardio performance. So when they will take, for example, EPO, these will have as final aim to increase their hemoglobin. And this is supposed to improve their resistance and their sport performance. So this is the final aim. But before that, all differences, red blood cells are supposed to increase. And what we like to call here baby red blood cell, the reticulocyte, the cells that comes before in the step of maturation of red blood cell will also increase normally.
Very interesting. And for people who might not be familiar with the physiology of creating new blood cells, how long does it take for these baby blood cells to be created? And how long does it take for them to mature into the adult form of the red blood cell?
So after we know around 1 week, so when we will take EPO, for example, after 7 days, we'll see the first result on the reticulocyte. And normally after 12, 14 days, we will have the result on hemoglobin.
Very interesting. And then for anti-doping purposes, why is detecting blood doping just so difficult?
So it's difficult because athletes are well trained to dope. And nowadays they take very micro doses, low doses with really small amounts of the doping products. So it's very difficult to detect it. And moreover, there are also different confounding factors, like the athletes that will go to train in altitude, also that they take iron that is not illegal. So this all thing together, it's coming difficult to detect blood doping.
Absolutely. And then just to give some context, what amount or what volume of blood or maybe EPO would be used when microdosing?
So about EPO intake, it's under the 20 microgram. So it's very small.
And do you see performance enhancing effects even at those tiny microdoses?
we don't see always there is the effect on the biomarkers. For this reason, I'm searching new biomarkers that can detect these microdoses. And about the sport performance, I will say it's more something that is in the mental for the athlete. They take the doping and they say, okay, we'll be better, but not really. It's more mental things.
That makes sense. And I'd love to dig into some of the details. So you're specifically looking at biomarkers, as we mentioned, for blood doping. So let's talk about these reticulocyte-related RNAs. So can you first maybe give our listeners some context? What are reticulocyte-related RNAs, and why are you particularly interested in studying these molecules within the context of detecting blood doping?
Okay, so these RNAs are the RNAs that are produced in the pathway of protein production in the reticulocyte. We don't know if everyone know that, but the red blood cells, when they're mature, they lose their nucleus and their DNA. But in the reticulocyte or the immature red blood cell, we still have DNA and RNA. So these RNA are supposed to increase when there is a blood stimulation, like doping, because these RNA are linked to the pathway of the red blood cell production and maturation. So we expect if we want to produce more red blood cells, this RNA will increase. So in our method to detect it, we expect if this RNA increase, red blood cell will increase. So if an athlete wants to increase these red blood cells, we should have an increase also in this RNA that are easy to detect.
That makes sense. And Francesco, can you walk us through your process a little bit in terms of what you went through to really launch and complete this particular research project?
So I come inside this project when I started in the anti-doping lab in Lausanne. So when I come here for my master's internship, the group here was already working around this RNA. And I just come in and go inside this project and then I continue to work on this project during my PhD. And now me and my colleague, Dr. Nicola Leuenberger, we are working on this, continuing on different projects, also in collaboration with other anti-doping labs and also in collaboration with other units from the more clinical unit to also see if we can take this project not only in anti-doping field, but also in a more large view with also other clinical sector.
Absolutely. And what was it about this particular project that caught your eye and made you want to pursue it?
If we stay in anti-doping, it will be to try to take this method that we are developing in Lausanne and also use it in other labs, anti-doping labs. And when we look about around clinical world, it's more, we already have a publication with the nephrology unit here in Lausanne, where we can see if the patient under dialysis that take EPO as a treatment could use this marker to follow the treatment if the treatment goes well. And also we will probably start a project with neonatology. So also to see the treatment of the newborn baby is always working.
Certainly. And I think being able to just take a step back and think about all of the potential applications of this project makes it particularly appealing. And you hinted at some of the potential applications on the clinical side, Francesco. I'd love to talk about those, whether it's dialysis and nephrology or potential applications in neonatal intensive care. So what are all of these potential clinical impacts of the work that you're doing?
I think it will help and could help all the nephrology because the EPO is used for dialysis patients to improve their red blood cell because endogenous EPO is produced in the kidney, but these people are kidney that are not working yet, are still working. So with these people, they are not really have a way to follow their treatment. They just look the level of hemoglobin, if it's increased or not. But if they're not increased, they're just giving more EPO. But with our method, we could see if the treatment is working well or if there is a resistance. And in this case, there is no sense to give more EPO. So with our method, we can see if the treatment is working well, also if there is a resistance. And also for the blood collecting, it will be easier because we need only, as I say, a dry blood spot to make the analysis. We don't need a blood tube. And also with the newborn baby, the premature baby that are very small when they're born and they need EPO to start their life, can also follow the treatment in these babies that are very small. And it's easier to collect DBS on this really small baby than blood tube.
Definitely. Well, it's exciting to see that this work has broad potential applications. And I'd love to dive into the details of the research. So your research, of course, showed that these reticulocyte-related RNAs from particular genes could potentially increase the sensitivity of the hematological module of the athlete biological passport. So, Francesco, can you talk a little bit more about how or in what situations this increase in sensitivity might occur?
So it will increase the sensitivity when we will speak about indirect detection of doping. when we speak about the athlete biological passport, especially in the methodological module, one of the markers of the module nowadays are the reticulocytes, but it has been demonstrated that the reticulocytes are not as much sensitive to the microdoses of doping. So we notice that these markers are more sensitive and will react after a microdoses. That is not really the case for the reticulocyte. So in this case, we could be more sensitive and detect microdoses of doping using this marker. That is not always the case using the actual marker that are the reticulocyte and the hemoglobin in the athlete's biological passport.
Very interesting. And beyond just enhancing that sensitivity, you're also finding that these biomarkers can increase the specificity of the ABP hematological module. So in what conditions or how are these biomarkers particularly valuable for really enhancing that specificity to particularly detect blood doping?
So it's more when we speak about altitude and hypoxic condition, because it's known that athletes used to train in altitude because there is less oxygen. So this could help them to improve their resistance and hemoglobin in a legal way. But sometimes this altitude will induce in the athletic biological passport some flag that are confounding flag because we could have false suspicion of doping, but these athletes are not doing nothing illegal. So our marker, we notice that they are very specific to doping and will not react to altitude, as is the case for the moment, for example, for the reticulocyte sometimes. So in this case, we will be more specific because we will be focused only on doping and not with confounding factors.
Very interesting. So in terms of implementation then, Francesco, do you see this as sort of a follow-up step that a lab would maybe take if a flag is raised in reviewing the athlete biological passport and they want to try to differentiate between doping versus maybe an altitude training kind of situation?
Yes, altitude situation. And also we notice that sometimes when athletes take iron because they have some form of anemia, it could induce some movement in the passport reticulocyte. And we already published a paper that we illustrated that our marker will not react to iron.
Well, that's really good news for the utility of this biomarker. And now let's, I guess, compare and contrast. I know biomarkers are a hot topic in anti-doping as well as other fields in science. So how does this method using reticulocyte-related RNAs as biomarkers differ from use of other biomarkers that you might be looking at or maybe other methods to detect blood doping?
With other markers, as I already say, we will be, for the actual indirect marker, like hemoglobin and reticulocyte, we will be more specific and more sensitive. Also, what is different with other markers that are already tested, like hepcidin, for example, is that this method is done by our method with RNA is done by PCR. That is a really well-known method, an easier method. And it will be also easy to use this method to have results faster in a fast way. That is more difficult with other methods. For example, also for blood doping, for the EPO doping, the direct detection is by using the Western blot, but it takes a few days to have the analysis. And sometimes we cannot detect the microdoses. So here we have a method that could detect microdoses in a quite fast way.
So in terms of the timeline, how long does it take to perform this particular test looking at the reticulocyte-related RNAs?
It takes around 4 to 5 hours.
Okay.
Everything from the RNA extraction from the derived blood spots to the final result with the PCR.
Definitely. And in terms of implementation in the labs, when you think about the traditional anti-doping lab, would they already have everything that they needed to complete this follow-up test? Or is this something where they would have to invest in new equipment and change their normal process to be able to implement it?
Some of them already have all the material that they need with all the machine also, because we already have some tests with other labs, with Paris, also with the Tokyo lab and Shanghai. So from their lab, everything is working. And I think other labs that we do not still not collaborate with them, but they already have PCR. So I think most of the lab are already in a good point to implement this method.
Very cool. And when it comes to cost, I know that's a big question in anti-doping as well. Is the cost pretty comparable to other biomarkers or other methods that you'd be using?
I think the cost is less than other methods.
Well, Francesco, I think this is a really cool project, and I think it has tremendous potential to have impacts. So when you think about the big picture, what are some of the potential impacts of your work?
I think it could impact a lot the biological passport of the athletes, giving a huge help to detect all these microdoses and provide more sensitivity and specificity. Also, all the tests for this method are done on dried blood spots, so that are easier to collect than the actual blood tube, EDTA tubes. So also it could increase the quantity of tests because it's really easy to collect just some blood drops from finger or from the arm with the new TASO technology. So I think this could impact on the number of tests and also the sensitivity and the specificity, as I said before.
Absolutely. And I know with dried blood spots, you end up with a pretty small sample and you sort of have to cut off little pieces of it to do different tests. Do you see this being a challenge where you would need more samples to be able to perform all of the tests you want to, including this reticulocyte-related RNA biomarker test?
Yes, of course, because only for this method, I need one entered dried blood spots, and I cannot do other analysis with the same spots. So yes, this could be sometimes problematic because we have to collect different spots to do all the analysis that we want.
That makes sense. And I think any new test is going to have some pros, some cons. And I think in this case, there are a lot of things going in favor of this biomarker test. So it is such an exciting development in anti-doping. We talked about how earlier this year you received one of the PCC-sponsored predoctoral research awards in collaboration with the American Physiological Society. And you actually had the opportunity to present results from your work at the annual APS meeting. So Francesco, what was that experience like for you?
So it was a really wonderful experience because it was my second big congress. I participated to the experimental biology last year in Philadelphia, but this one in Long Beach was way better because it was really only the physiology. And it was for me the occasion to meet new people from all my network, speak with a lot of people. And all the congress are really well organized, all the scientific part, but also all the social part with different events. So it was a really cool experiences. And it was also for me my first time in California. So it was really nice to discover this region and this state.
Very cool. And in terms of presenting the work, how was it received? Did you get a lot of good questions and a lot of interest at your poster?
I had a few questions, few people that are interested about my work. Of course, there are a lot of physiology congress and with different field from physiology. And anti-doping was not the biggest one, I think. All of people are there from maybe renal physiology, cardiac physiology. But when I speak that I'm working about in anti-doping, all the people are quite interested because it's something that we not know a lot and people that are not in the field maybe don't know a lot about what we are doing here. So yeah, people were interested in my work.
Excellent. I'm glad to hear the work was well received. And I'd love to talk about next steps, Francesco. What are the next steps for you in this line of research?
For me, the next steps will be, first of all, finish my PhD. So I will finish only next year, in the beginning of the year. So now my next step will be to write my final thesis, prepare my oral presentation for my PhD defense, oral defense, and also trying to finish or go above with this project in anti-doping and also with the different clinical fields and present also this project as more people as I can.
I think that's wonderful. And I know in anti-doping, it's a very multidisciplinary field. So you have people coming in from analytical chemistry, biochemistry, biology, physiology, medicine, psychology, social science, ethics, and just a variety of other fields. And from your perspective, Francesco, what do you think physiologists can contribute to the conversation? And why is it so important that we have physiologists involved in anti-doping science?
I think physiologists could help anti-doping with all their knowledge about, for example, altitude, hypoxia, because in anti-doping there is all, as I said before, that lead training altitude, and hypoxic physiology could tell us more how the different biomarker will move in case of altitude. Also with the renal physiology, because we'll have this link, as I say, with nephrology and all the dialysis person. Also with cardiac physiology, because it's known that if there is too much abuse of different products, that could induce cardiac problems. So I think all this work, all these physiologists could help anti-doping, and anti-doping could help them. And also sport physiology, because we could make link between the real sport performances and if there is an effect or not from the doping product.
Absolutely, Francesco. You brought up some great points and examples there, and I'd love to see the number of physiologists in anti-doping continue to grow so that we can leverage everyone's different expertise to answer some of these tough questions in anti-doping. But of course, when answering these difficult questions, there are challenges. So let's talk about maybe some of the challenges that early career researchers may face as they enter the field of anti-doping, or maybe just in science in general. So, Francesco, what are some of the biggest challenges that you've faced as an early career researcher?
So, biggest challenge, I think for the moment, everything goes quite well. Sometimes there is some difficulty with sending some samples or receive the samples in time, but for the moment, everything is going quite well. And yeah, maybe the big difficulty is to prepare the paper, then after research, put all the results together, the analysis. Maybe the quantity of work is challenging sometimes.
Absolutely. I think it's a long process sometimes in science from sort of the idea of when you want to do this particular project to kind of laying out all the experiments, doing all the work, getting everything written up and finally published in a journal ultimately to see that final product. But what are some of the biggest successes that you've had so far?
So the biggest success, I think, is the different publication and also the fact that I could participate to this congress that I'm not focused on anti-doping, but also in other fields. And I think one of the biggest success is the PCC award that recognizes all my research. And I am very grateful to the opportunity to win this project. And this gives me the opportunity to go to the congress in Long Beach.
Definitely. Well, the PCC is excited to support you and your work. And I know there are often barriers that prevent you from answering all of the questions that you'd like to answer in science, and whether this is just sort of time, feasibility, and the amount of work in some cases. But if we gave you all of the resources you could dream up, Francesco, is there one problem in anti-doping or one question that you would want to solve most?
If I can have all the resources imaginable, it will be to be the best in anti-doping research, and that anti-doping is going always better, and stop doping at all, I think.
Is there one sort of dream project that you have in mind that would take you a step closer to that ultimate big dream of stopping doping altogether?
I think, maybe my project will give a little step. So, this dream, but I think it's extremely difficult because there will be always new technology for athletes to dope, and it's always a fight between laboratory and anti-doping association against athletes that find always new methods and technology to dope.
Absolutely. I think sort of getting a window into what's going on with the athletes so you can stay that one step ahead, I think would be a huge step towards getting there.
Yeah.
Well, Francesco, you've done a wonderful job sharing your work with us today. If our listeners want to learn more, what is the best way for them to do so?
They can find all my research on PubMed, because all my publications are there on PubMed. And also they can find me on LinkedIn.
Well, listeners, definitely get connected with Francesco on LinkedIn. Check out his papers on PubMed and read up on the amazing work that he's doing. And Francesco, thank you so much for joining us on the show today. It's been a pleasure to chat with you.
Thank you. It was a pleasure also for me. Thank you to give me the opportunity to have this interview. And it was a pleasure also for me.
Wonderful to have you here. And listeners, great to have you here with us as well. If you haven't already, it would be wonderful if you could rate our podcast and leave a review on your favorite platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.